Hello, everyone, and welcome to the Human Cell Atlas Biological Network seminar series. Today, we're focusing on spatial omics. I would like to remind you to please join the Human Cell Atlas if you're not already a member. And when you do join, you can also join any of our 18 biological networks. There are multiple ways to get involved. Please take a look at our website, and we're happy to welcome you. I wanted to also remind you of the upcoming Human Cell Atlas events. The HCA Asia meeting is happening in December in Hong Kong. The HCA Town Hall is a virtual event. We're hoping to see you on January 23rd. There will be some exciting announcements there and a state of the HCA talks. So we're looking forward to having you there. The HCA General Meeting in 2025 is in June in Singapore, and the registration for this will open in early December. Please stay tuned for announcements and emails from us. We're always welcoming meeting sponsorship and exhibitor opportunities. Please do contact us at meetings@humancells.org if you're interested. Another exciting announcement is please stay tuned. Next Wednesday, the HCA Nature Portfolio Collection is going live. There will be many announcements, and we're very excited to have more than 40 papers in this collection. And please stay tuned for future collections as well. Meanwhile, if you have a publication that you would like to include in the HCA branded publications, please visit our website and see how to go about it. Doing this will give your paper more visibility, and please inquire about how to do it if you have any questions. Today's seminar agenda, our discussion moderator is Holger Heyn, and our speakers are Anna, Ilya, and Arutha. Please submit your questions using the Q&A function in Zoom, and we will either address it during the discussion or the speakers will respond in writing. With this, I would like to introduce Holger Heyn, who is a professor and leader of the single-cell genomics group at the Spanish National Center for Genomic Analysis. His group combines technology development with research activities that focus on cell atlas projects and clinical immuno-oncology. Together with Orit Rosenblatt-Rosen, he co-chairs the Human Cell Atlas Standard and Technology Working Group. And since Holger moderated the spatial omics session at the HCA General Meeting in Milan in September, we asked him to summarize what has been discussed there and also to moderate today's webinar. Holger, welcome.
Thank you very much, Ellen. Do you see my second screen and the presentation now?
Yes.
Okay, thanks again, Ellen. And it's obviously a great pleasure to host this spatial webinar today and to moderate the panel discussion at the end of the hour after the talks. But let me take a step back to really give you insights why we selected spatial as a topic for this webinar today. It is mainly because as a community, we feel that spatial technology is ready for prime time. That means it is robust enough, it is scalable enough, for example, to generate atlases of the healthy human system. And that's where we come in, and because the HCA, so the Human Cell Atlas, has always been built on two pillars. One is the dissociation single-cell sequencing based methods to generate atlases, but also from the beginning, from inception. On the roadmap, we had spatial atlases generated by spatial technologies. And now, during the Milan meeting, the annual meeting this year, we discussed the initiation of a new program, potentially a spatial program, where we then gear the focus towards the generation of spatial atlases, the integration of single cell, what we have now, together with spatial initiatives, really to go beyond a UMAP, and then for the future, allow the community to browse cells in tissues in 2 and potentially 3 dimensions. So here we really envision the kind of implementation of such program as a multi-step process, where over the last almost 10 years now, we have been generating single-cell resolved atlases using dissociation-based methods. And that really allows us to produce a first version of human atlases, many of those already published, many of them being published as we speak or being generated. But now it's the time and technology is ready where we really move from single cell to the spatial context. And there are many integrative efforts already being done where single cell is combined with spatial. But moving forward, we really aim to go deeper through these levels and to profile human cell atlases with spatial technologies alone, initially in 2 dimensions, later in 3 dimensions, really zooming through tissues and organs, but eventually also include a kind of temporal component where we then map cells in organs through time and space. And this really means, as an ultimate goal, is that we're not working on dissociated cells anymore. So we're not taking cells out of their natural context, but we're also not slicing cells. So current methodologies really slice cells into smaller sections around, for most of the message, 5 micrometers, which really gives you a kind of snapshot of the transcriptome or genome profile that is represented within the cell. But eventually what we want to achieve as a community is to zoom through cells in 3 dimensions and eventually to zoom through tissues and organs in 3 dimensions. So that's why we discussed and we plan the implementation of the spatial program, which we envision to structure in 3 different tracks. The first track mainly focusing on the technology part, so technology scouting, benchmarking, implementation, and then having a second track focusing on the data analysis, starting with data storage, processing, and obviously most importantly, the visualization and analysis with the interpretation. Most importantly, though, is eventually that being funneled into the generation of high quality spatially resolved atlases for the healthy human system, but also now as a community, we move from the healthy towards the inclusion of diseases to also include the kind of human perturbation aspect. So here we are obviously not starting from the scratch, but we are standing on the shoulders of giants, really working with the already established working group. So we have, for example, the Standards and Technology Working Group that I'm co-chairing, that is massively then involved in sharing the expertise together with the first track that is focusing on technology. But then we have the Analysis Working Group, but also obviously the Bionetworks that gained over the last years a lot of experiences in their respective organs that will then help us making wise decisions, what technologies to choose and what projects to aim at first. So here, this webinar today is really structured in the same way that we have talks related to technology, analysis, and atlasing project. And we have champions for each of these tracks that will allow us to kind of guide through the, for example, technology selection process, examples for algorithms or analytical pipelines, including cell segmentation. And then we have one talk very much focusing on the translation of spatial technologies into the clinical context. So here, one of the main challenges still remains that the technology landscape is still very diverse, which obviously is positive because we have many technologies to select from. But here, as a community, really work a bit on this guidance system, what technology to use when. And for spatial technologies, we can broadly split approaches into sequencing or imaging-based methods. For sequencing, molecules are captured from tissues and then subsequent analyzed through next generation sequencing mostly. And then we have imaging-based methods where molecules are directly imaged or profiled in tissues, which then allows us to profile molecules at molecular resolution, put into cell segmentation algorithms to resolve spatial profiles at single cell resolution, but then other methods work on pixel level, where then the size of the pixel is very much defined by the size of the underlying capture units for sequencing methods. And here for most of the projects, we today focus on RNA and using RNA, for example, for the phenotyping of cells, but there are already many, many methods out there that implement different modalities, including DNA, protein, and chromatin, and many that combine some of these modalities into a multimodal readout. And really, it is us now as a community to choose and to pick what is the best fit for purpose for our given project. And here, as a program, we will work very closely with the biological networks to look at the needs for specific organs and then choose from this catalog of different technologies the best methodology that is best fit for purpose for a given question to answer and to allow informed decision-making processes. And today's webinar is very much structured in the same way. So we have Anna Pascual talking about the technology aspect and describing new technologies that are kind of cutting edge, pushing into the area. And we have then Ilya Korsunsky very much focusing on the computational part. So Ilya comes from a strong single-cell computation background. Many of you will know him as a developer of Harmony and Symphony, so probably one of the most used integration tools out there for single-cell. And he has also been working very hard on kind of generating tool sets for the spatial context. And then we have Arutha wrapping up, really sharing his experience, bringing spatial technologies into the clinical context with a long experience, how he brought spatial methodologies to clinical kind of great profiling. And with this, I will already hand over to, stop sharing my screen first. I will hand over to Anna Pascual, who's joining us here from Barcelona, from the CENAC. Anna joined the CENAC as a team leader for spatial genomics, coming from the Charité in Berlin. We are very happy having you here. And I'm very happy to have you in this webinar today, and very much looking forward to your talk.
Thank you,
some
Holger.
more housekeeping though. Please ask questions in the chat. So Anna will answer those questions just after her talk, and some questions we will bring into the panel discussion at the end of this hour. So Anna, your stage. Take it away.
Thank you very much, Holger. So I will be sharing a glimpse of a study in which we are using whole transcriptome single molecule imaging to map the emergence and progression of colorectal cancer. And for that, we are using two volvulus adenoma samples, which are pre-malignant epithelial lesions, so polyps, that are known to be precursors of colorectal cancers and to give rise to both mucinous and adenomatous cancerizations, even in the same sample and patient. But it's not yet fully understood how molecularly this happens, and what we want to map is how healthy mucosa transitions to the pre-malignant lesions, and from that to the tumor core. So as a pilot, what we did is to combine different data modalities in two samples. The one in the left, in which we have normal mucosa and the TVA regions, distinct TVA regions, and then the other one, the one in the right, in which we have normal healthy mucosa, if we can call it that way, of course, TVA regions, and two different types of tumors. So we have the H&E to define these distinct histopathological features, but then the sequential tissue section we put under the CosMx instrument with the WTX assay, in which we have 4 proteins and 18,000 genes approximately as readouts, with more than 500,000 cells per sample. So here the aim is also to assess how good the data is, especially since this assay is not commercial. So we want to test if we can actually extract the biological complexity of the tissue and the disease. And on top of that, and from the next adjacent tissue section, a thicker one, though, we will also get a richer single-cell RNA sequencing data set that will be processed, or that will be generated with PathoSeq protocol using the Flex kit from 10x, and that we will use as a reference data set. So for each tissue, the 4 proteins that I've mentioned that are acquired from the same tissue section will be used on the flight in the Cosmics assay to generate cell segmentation masks. And then the single molecules imaged, and this is just one example of OLFM4 molecules. Each white dot is now a molecule. These molecules will be assigned to individual segmented cells, and with these we can generate this matrix composed of cell IDs, molecule counts for each of these 18,000 genes that we have in the panel, and then their spatial coordinates, so that we can perform clustering analysis and cell annotation in situ, and then all sorts of downstream single cell spatial analysis, everything we can imagine. So how do we perform clustering analysis? Actually, we used the in situ type algorithm, which was developed by NanoString and specifically tailored for Cosmics data. So it uses not only the counts matrix, but also the background signal, the immunofluorescence staining, and also parameters such as cell area and shape. So there's no principal component analysis or similar. And you can run this fully unsupervised, looking for free cluster profiles, and then assigning cell types by probabilities. Or you can also use it in a semi or fully supervised mode with a reference data set. And that's exactly what we did with the single nuke patho-seq from the same tissue blocks. So, and what we do after the first clustering round is to pool the clusters in 3 main compartments. So epithelial, stromal, and immune cells for each sample separately. And then we recluster each compartment independently in a sequential step to actually resolve better the heterogeneity or complexity within each of these compartments. And I will now focus for a while on this particular sample, and let's start with epithelial compartment because it's beautiful to see how the transcriptional data actually recapitulates the histological features. So while we have steady state epithelial populations that now for simplicity, let's say, are shown here as normal 1, 2, and 3, and they only appear in the normal crypt areas, which are annotated as N1, N2, and 3. They are completely absent from all TVA regions. And then we have some TVA regions, TVA 2, 5, and 4, which are marked by a transitional epithelial cluster, this sort of cyan, that shows an upregulation with both differentiated and undifferentiated genes. And then there are some other regions, TVA 1 and 3, basically, that are composed of stem and fetal-like clusters, which are particularly obvious in this light green region called TVA 3.2. Which is a villus-like structure as annotated by the pathologist, which has a very unique transcriptional profile, but also a very unique sort of morphology. It grows upwards, away from the basal membrane and towards the apical side, similar to what villi do in the small intestine. So that already caught our eye from early on. And since we can recapitulate very nicely the histopathology with the transcriptional data, then we can use ROI annotations to actually perform pathology-aware analysis. And this actually helps quite a lot on interpreting results. So that's what we do. And then we can focus on particular regions of interest. For example, if we look at this one, we have a real transition in a, let's say, spatial continuum from normal crypts in the right side to transformed premalignant epithelial lesions in the left side. And we can easily observe, actually, that there's a very distinct abundance in immune cells, for example, very abundant on the normal lamina propria and not that much abundant on the TVA region. But not only that, there's also a different distribution of immune subpopulations in the normal, which are basically characterized by IgA plasma cells shown in pink colors in this bar plot, versus the TVA regions, which show higher proportions of affected T cells, for example, or tissue-resident macrophages. And the arrows here point to these regions of interest that are shown on the site. And actually, the same happens with the epithelial compartment. Normal areas show similar composition as expected, and then we have blocks of TVAs, 2 distinct blocks of TVAs that behave similarly. But then we have this ROI, this TVA 3.2, again, that stands out particularly, which is very different from the rest. And that corresponds to the villus-like structure that showed this apical growth and that I've pointed to already. So since we have these different morphologies also at the tissue lesion, at the tissue level, we also wanted to perform some type of quantification of the morphology, really. And what we did is to extract morphological metrics at the crypt level, so taking crypts as units and doing that at the tissue-wide scale. And whatever we look at in terms of morphometrics, they do separate quite nicely the normal epithelial regions or the normal crypts from the TVA regions, which links back to the compositional and the transcriptional differences. We can also look at signatures to sort of calculate whether a critical mass of the input gene set is actually enriched. And since we saw that in the epithelial clustering, that the most transformed tissue areas were composed of stem-like or fetal-like epithelial populations, we can plot 3 independent fetal signatures from the literature across histological regions and across epithelial clusters. And we actually see that they are enriched in the fetal populations as annotated broadly, so validating these preliminary annotations. We can also map them back on top of the tissue, let's say, and see the image that this gives us, which ends up again in this villus-like structure in the TVA region 3 at the very end. Okay, so now taking a region of interest composed of pure, healthy, normal epithelial crypts, let's say, which is also very nicely cut longitudinally and represents the basal to luminal axis, along which the epithelial cells are known to differentiate from stem cells towards fully differentiated colonocytes of both the secretory and the absorptive lineages. This is actually the perfect scenario to test trajectory analysis. We used Slingshot for that and see if this would reconstruct this well-described epithelial differentiation gradient. And this actually maps the pseudotime along this crypt axis very nicely. So now that we know that we can recapitulate what's already known about normal epithelial cell differentiation, we can apply it at a tissue-wide scale, and this would allow us to infer transcriptional trajectories in space and time. And that's what you look at. So the yellow is later pseudotimes, which again bring us to the villus-like structure. And we quantify this. The pseudotime recapitulates this progression from the normal epithelial regions via this transitional epithelial cluster and to the stem and fetal-like populations shown in orange and green. And now we can also try to explore how the epithelial niche, which would be the stromal and the immune cells that sustain and interact with these different epithelial cell states, how they also change along the pseudotime axis. And we detect a transition in the stromal compartment along this pseudotime from normal fibroblast to CXCL14 pro-inflammatory fibroblast, while, for example, in the immune compartment, we come from a lamina propria, let's say, enriched with IgA plasma cells, as I've pointed before, and then towards an enrichment with exhausted T cells or tissue-resident macrophages in the later pseudotime, which again map to these stem fetal epithelial populations at that very particular region of the tissue. So with these 2 samples, let's say we we have actually, we are quite convinced that we can get a nice resolution of different cell states in all compartments in these tissues. And that these aligned to pathology, we can actually plot pathway signatures. We can perform pathology-driven analysis, and we can plot spatial gradients and even trajectory analysis in space. But to try to further understand these transitions, we need more samples. So that's actually what we are on at now. So we acquired 6 additional samples using the same assays and data modalities. And that's that's ongoing work, right? But we have also this sample 222, that does not have any tumor lesion. Similar to the one that I've been talking about, all the rest have annotated tumor lesions, and they are actually diagnosed colorectal cancer patients. So what we are doing now is to integrate the information across all sections, also with a matched single nuke path sequencing data. And we are performing cell-to-cell communication, niche analysis, etc. We are doing that with more than 3 million cells in total. That's the data set that we have as of now. And it's already interesting because these 2 samples that do not have any tumor lesion identified, when we pulled all data together and ran the same type of clustering analysis, but now with the integrated object, let's say, it stands out already that in this villus-like structure, we have this dark blue cluster. This dark blue cluster that appears in any other sample that actually contains tumor lesions. And if we plot these different blue clusters from each sample in space, they are indeed localized within the annotated tumor lesions by the pathologist. And the other only sample that was actually an FPA patient, not a CRC patient, does not have this cluster. And we are now really looking into that, sort of looking at what genes actually characterize this population. But they are basically related to cell proliferation and to cytoskeleton rearrangement. So maybe aiming at cell adhesion and migration, helping on tissue displacement and invasion. We are looking into that more in detail. We are further analyzing all these results, and we'll hopefully be able to show them soon. And so I will just now finish by acknowledging the people actually involved and driving all this work. Holger, whom you all know already, and also Helena, which is an extremely talented postdoc that has actually performed all the analysis that I've been showing and generated all the visualizations. And then also Irene, which was the last addition to the project, and that is crucial in annotating all the samples and interpreting the results. So, and also thanks to the rest of the team, Max, Patri, and we love to play with single-cell spatial technology. I want to use the chance to actually say that we are hiring. So if any of you is looking for a new position and wants to have fun with us, please get in touch. Thank you for your attention as well.
Thank you, Anna. And I can't agree more. We love to play with single-cell and spatial methods, and that's what we're doing in the end. So thanks for the wonderful presentation. And for Anna, please ask now questions in the chat so she can answer right away by writing, but some questions we will take into the Q&A panel discussion at the end of this session. And with this, it's my pleasure to introduce Ilya Korsunsky, who's joining us from Boston, who has a group at the Brigham's and Women's Hospital in Boston, together with Harvard Medical School and broad appointments. And his group is mainly focusing on generating single cell, but now also spatial profiles for autoimmune diseases, working on immune tolerance, infectious disease, but also on cancer. As you all know, he's the mastermind behind Harmony and Symphony. And I understand he's now applying his knowledge in the spatial transcriptomics and genomics field. So Ilya, very much looking forward to your talk. Thanks for agreeing to present today. And yeah, the stage is yours.
All right. Thank you so much, Holger. Thank you very much, everyone at HCA for inviting me. Yeah, it's a very vibrant community, and happy to be a part of it, and see what we can contribute. So today I'm going to talk about, as Holger said, some of these recent ways that we're thinking about analyzing single cell spatial data. But before then, financial disclosures, lab funding, personal consulting. I'm going to do a quick introduction to my lab so you guys know all of the people actually working on these projects. And you can find out more about them at our website, korsunskylab.org. But basically, our research kind of boils down to these three major categories. We want to understand how immune cells, stromal cells, act in autoimmune and inflammatory diseases. We want to understand how they act in the context of cancer immunology. And we, of course, use single cell and spatial transcriptomics to answer these questions. And of course, we then develop methods to be able to analyze these data more robustly and get more biological insights from these data. So these are a few of the representative papers from the group. Of course, none of this is possible without a really broad range of collaborators. I'm showing some of our more, some representative collaborators from each of these areas here. We're really responsible for generating the data and together, generating ideas. So I'm going to start with my very biased personal opinion. I think spatial transcriptomics is so cool. And this is not a collaborator here, but this is Captain Obvious, I think, to everyone who's attending here. But why I think it's really cool is that it's just so we can learn, we can infer things with statistics and then go back and see it. And that's so unusual coming from genetics and even single cell transcriptomics space, where we have these very abstract things. Now we can go in and actually see transcripts in space, see cells in space. And I can't tell you, I've spent hours and hours just browsing like the VizGen website. They put up their public MERScope data sets, and this is so beautiful to me that you can look at two genes. Every dot here is an individual transcript, and you can see the outline of fibroblasts. You can see the outline of blood vessels in green. You can see cells that have nuclei, holes in the middle here, and it's so palpable, right? And the technology that we're looking at nowadays, we can do this not for just two genes, but hundreds or thousands of genes. And so the best thing is that now we can take all of our knowledge from single cell, transform it into spatial, and we can see all of our cells in space, right? So this is an unpublished study, collaboration with Dr. Wei's lab, led by Rupa in postdoc Rupa Madhu in our lab. This is in RA. We can identify, we have, something like 2 million cells from inflamed synovium, and we can see it now in tissue. This is one little representative piece of tissue. And we can see in brown, we have blood vessels. In blue and red, we have immune aggregates. In green and purple, we have the lining layer of this tissue. And this is so cool. It's very exciting when we see it. But then one of the hardest things is that when we say, wait, well, we have thousands of genes. Why limit ourselves to 10 cell types, right? And so we worked really, really hard, and we figured out some things. And we said, okay, we figured out how to label 50, 70, 100 cell types, 100 cell states, transfer over cell states from single cell into spatial. Now we can see 70 cell states in tissue. Now it becomes not just cool, but it becomes really hard to look at these, right? Because there's just too much. There's too much stuff going on. And so being able to take tissue and automatically organize it into niches, regions, zones, anatomical whatevers, it helps us really be able to handle that complexity, right? Then we can zoom in on one region and say, okay, this region has 5 cell types. Who's talking to each other? What are the key cell types? What are the genes they have? And so we've been thinking about this problem actually for a while, because even before high-dimensional spatial transcriptomics, we've been using other technologies. In 2021, we had a paper where we looked at fibroblasts across 4 different tissues, and we used, I think, like a 25-35 color proteomics assay, and we used this approach to try to automatically find tissue regions. We said, okay, if you take a cell, don't cluster it by its internal protein expression, but cluster it by the expression of its neighbor's protein expression. Now you're clustering neighborhoods instead of clustering cells. Well, we weren't so clever. Lots of people thought of this. In fact, this is a paper from around the same time for this UTAC method. They do the same thing. And we were pretty happy with their results. We were able to get major structures like immune lymphoid aggregates, perivascular niches, vascular niches, fibrotic niches. And if we compare the raw data to the niches that we called out algorithmically, we were pretty happy. And it was good for the purposes of our analysis. Then in 2023, working with Nir Cohen and John Chen, this is our first foray into cancer immunology. We already knew from our pathology experts that we were looking for this particular immunity niche that was about 50, 100, 200 microns in length. And so we took an even simpler approach and just said, okay, what if we just grid the whole tissue, get all the transcripts in one grid, collapse that, and then just cluster those grids. And that worked really well too for us, actually. We were able to identify major anatomical regions. We were able to identify all of the different immunity niches that we were interested in. But we started out with a very targeted hypothesis, right? But what happens when you zoom in and you're not interested in broad picture structures, but you're actually really interested in very fine-grained structures where the boundaries really matter? So here's a cartoon I drew of some arbitrary cells. NIH just recently released this repository of cartoon cells. I didn't cite them, but this is from the NIH recent portal. So what if we have these cells on the left that are clearly different from all these interacting cells on the right? And we don't want to treat them as one region, right? And so if we take a tiling approach, well, that's not going to work really well, right? Because there's no guarantee that the tile boundaries are going to actually cross the natural boundaries of the tissue. And in fact, the neighbor smoothing approach is better, and it works sometimes, but still, if there are cells at the boundaries, they're going to borrow information from everybody, not just the cells in their compartment, but cells from neighboring compartments as well. And so we get these blurry boundaries or no boundaries at all. And so we've been working on this problem, and we come up with an algorithm. This has been work led by a PhD student in my lab, Daniel Stein. He named it Tessera, which we're gonna introduce this into the genomics lingo. This is the actual name of the tiles that are used to make like these ancient Roman and ancient Greek mosaics. So it's the name of the tiles themselves. So what Tessera does is it pays attention to the natural boundaries of the tissue, and it draws tiles that agree with the natural boundaries of the tissue. So the preprint is forthcoming in a few weeks. We're very, very close to having something we're ready to release. But the code is actually up right now, and the package is ready to download and use in R. And I'm going to walk you through a little bit of how it works. So the first thing is we take cells in space. So every dot is a cell, and every cell is represented by, let's say, its PCA coordinates. And when we zoom in, we see that, okay, this PCA embeddings, which is just a compression of gene expression, it has no spatial information at all, right? But when we look in space, there are spatial patterns, right? So there's this strip of blue cells, and then there's a strip of red cells. And so this gene expression in space has information. And in fact, we have this for 20 PCs, so every single PC gives us some information. And so what we can do is we can say, okay, every cell, look at your local neighborhood and look at the gradient of gene expression changing in XY space in your local neighborhood, right? So the cell we circled right here, all the cells on the bottom are like light blue, all the cells on the top are reddish. And so there's a large gradient going north to south. For these other cells, similar thing, but for the cells in the middle, who's, if I'm a cell, all of my other cells have roughly the same PCA values, there's no strong gradient there. And so what we get is a gradient field over all of the cells in the tissue. And it's pretty messy, actually. And so we use anisotropic smoothing, which is basically asymmetric smoothing, to smooth it out. And now what we get is actually pretty cool. we get some regions where the lines are really small, and that means the gradients are really small. That means gene expression is not really changing. And so that's probably the inside of a tile, the inside of a region. And then we get regions, and then we get these very consistent borders, these consistent edges between things. And that's probably the boundaries of regions. And we want those to be the boundaries of our tiles. And so what we do is we compress this information into just saying, are things changing at this local position, or are they not changing? And so if it's blue, if it's not changing, it's yellow if it is changing. We can do that for the whole tissue. And then we can basically draw lines using this tool. It's called discrete Morse theory from topological data analysis that basically tracks the yellow, walks along these yellow gradients here. And then we use agglomerative clustering to group together similar tiles that are adjacent. And so I'm going to go back and forth between these two a few times. We simplify the picture a lot into these nice tiles, some anywhere between 1 to 10 to 20 to 50 cells, depending on the parameters you use. And they agree with the gradients that we found in the first step, where we found the natural gradients of the tissue. And so what you get at the end, if you go ahead and plot your cell types, is you get tiles that contain many cells, but their boundaries respect the natural boundaries of the tissue. And so this is it. This is the Tessera algorithm in a nutshell. Again, this is Daniel Stein's work. You're gonna see the preprint up soon, and this is the algorithm in a nutshell. So where does Tessera actually shine? So we've tried it out in a lot of different situations. The one where I think is nice to present, and this is this recent publication from Zhang et al. In Nature, where they took whole mouse brains and diced them up into 100, 150 different slices and did 1,000 gene merfish on every single slice. So very rich information, 4.2 million cells here. And so we took one of these coronal sections, and they already provided previous cell type annotations here. So we kind of know what to expect. And then we run Tessera, and we get these Tessera tiles. And then we can treat each of these tiles as a unit of analysis, cluster them, label them. Cluster them, label them, and then get cool things like blood vessels in gray here. We can call out ventricles here in the middle. We can call out we can see hippocampus here. So we get these very specific structures that we expect. And they don't not necessarily lie on very straight edges and straight boundaries, because that's just not the boundaries of the actual tissue. And so then we asked, how well are we doing? And we did some benchmarking against other state-of-the-art algorithms. And it turns out that if we look at really big regions, so like cortical layers of the brain, L1 through L6, we do pretty well, and everyone does pretty well, actually. These are very regular regions. Even at the boundaries, if things change a little bit, the overall picture doesn't change very much, right? And these boundaries are honestly, they kind of bleed into each other, maybe naturally, one could say, right? Sometimes we label L5 neurons, L6 neurons, but then we have like L5, 6-ish neurons. And so these are kind of, everyone does pretty well on these large structures. But where we really see the differences is when we start to look at smaller structures, especially ones that have thin boundaries or irregular shapes. So here's two examples of ventricles from this one mouse brain slice. And while Tessera captures the boundaries of these ventricles pretty well, there are some cells that are colored outside of these black outlines, and that's where we made a mistake against, ground truth annotation. But overall, we don't get too many cells outside. But algorithms like Stagen and Cell Charter, they use this isotropic diffusion idea, right, where I borrow information from all my neighbors. And so what happens is that I'm going to inevitably blur my boundaries. And then I don't get this very clean separation of this one particular structure from its neighbors. And then the cool part is we can apply this to the whole brain, and it's just a beautiful picture because this is now all 147 coronal slices, left to right, top to bottom, going from anterior to posterior, and we can actually see how the regions change. So the colors here are actually colored by Tessera tile clusters, and we can see the type of structures change very smoothly. And this is also our chance to see how well we do in terms of computational scaling. We do pretty well. This is over 4 million cells, 150 batches. Using only one core, it takes about 30 minutes. It's inherently parallelizable, so we think we can get it down to 2 or 3 minutes on 4 million cells. And then we get this nice picture of how things change across time. So that's pretty much it. So we do have a preprint coming up very shortly at the beginning of December, see it on bioRxiv, where we have more exciting examples of large patient cohorts in brain, in lung cancer, where we do xenium 5K analyses to show how we can actually trace out germinal centers in lymph node, and we can actually order them by early to late. And if you're interested in this, yeah, you can try it out yourself. The package and the vignettes are actually all online on GitHub, open source. And yeah, if you ever want to come visit us or come talk to us, we're very happy to take visitors. So thank you very much.
Thanks, Ilya. I agree. Spatial genomics is very cool. I like the opening, and yeah, congratulations. Very much looking forward to the discussion later. Talk about area profiling, cell segmentation, single molecule resolution. And now I'm very honored to introduce Arutha Kulasinghe, who's joining us from Australia. And I know it is very late in Australia. So thanks for staying awake, for staying up. So Arutha is an associate professor and leads a clinical omics lab at the University of Queensland. And he has been the pioneer of spatial genomics in the Asia-Pacific region. He's mainly working on pathobiology and is working on kind of integrating multi-omics layers, including spatial genomics. So Arutha, I'm very much looking forward to your talk, and thanks again for staying up. We owe you coffee.
Thanks, Holger. It's hard to say no when you get an invite from you. So let me know if this is coming across on your screen.
Yes, not full screen though. No, perfect.
Okay, brilliant. Thanks, Holger. It's wonderful to be here. It's 2 a.m. in Brisbane, and so some of it might be a bit slow. But anyway, it's to give you a flavor of our applications using spatial proteomics, spatial transcriptomics in applications of sort of clinical challenges in IO. We've acknowledged a number of funding agencies that support this work, both nationally and internationally. So one of the biggest challenges for checkpoint inhibitor therapies is that, and also ADCs, is that the lack of predictive biomarkers. This work's really led in the field by what's been done in melanoma, non-small cell lung cancer, triple-negative breast, head and neck, and so on, where about 15 to 30% of patients respond to these therapies. And so this article came out a couple of years ago. It was a commentary in Nature which showed that tissues and not blood are where immune cells function. And so I did my PhD in the liquid biopsy space. And so I realized quite early on, we need to go back and map the tissue context and get that visual information simultaneously with your molecular readout. And so there are a number of predictive biomarkers for checkpoint inhibitor therapy, PD-L1 expression, which is sort of a single marker expression, and also tumor mutation burden, which is sort of a bulk measurement of your sort of neoantigen load. What my lab tries to do is to map the entire tissue, sort of context, just 3 to 5 million cells from surgical resections, from biopsy samples, and to really understand every cell that's in the composition of the tumor and the stroma, and also the interfaces. And so I don't need to tell this audience why getting that visual data simultaneously with the molecular readout is important, but sort of long story short, when you have a high degree of immune cell infiltration to the tumor, these tumors tend to do better with immune checkpoint inhibitor therapy because you've got activation and engagement of those immune cells with the tumor. But if you were to look at them at a bulk level, you'd get very similar TIS scores. So the tumor on the right is more infiltrated compared to the one on the left. And so I like to talk about technology stacks, and this is what our lab uses. Initially, we did spatial transcriptomics up front. We'd go and validate using spatial proteomics and sort of other approaches. But what's happened over the last 2 to 3 years is as the plex levels have increased on the protein end, as we've been able to get 50, 60, 70, 100 proteins, that potentially becomes discovery too in how you develop those panels. And so the computational bit that comes together here is we work with multiple partners across the world, both in AI, ML, tissue biobanks, to really sort of modularize some of the pipelines for large cohort studies, and so flagged a number of partners we work with here. And so these are sort of the major studies that are happening in the lab. In lung cancer, we're looking at about 300 patients, multi-site. Multi-site is fundamental. A lot of these studies are single-site studies, both retrospective and prospective, and we've got really strong initiatives in lung, head and neck cancer, skin cancer as well, where we're getting tissues over the course of immunotherapy at biopsy, on therapy, at cure, and at progression. We get tissue punches, and at cure and progression too. And so we can look at the temporal changes in the microenvironment. And we've got a number of breast and colorectal studies. The colorectal is really interesting. It's 50 tissue microarrays over 50 slides. It's a 2,500 patient cohort of 10 years of annotated tissue. So we've got activity across all these spaces using multiple spatial genomics and proteomics approaches. I'll walk in some of the work we do. So we work in both tissue arrays and whole slides. This was one lung cancer study where we ran a number of platforms, but we'll talk about some of the proteomic findings. This was single core biopsies across about 50 patients, second line IO versus standard of care. And so we built this panel focusing on the immune cell profiling core, various modular cores that we can put together, but we added in custom markers which give us functional activity over phenotype information. And so we can identify the subsets within each individual cell type that might be playing a role in response or resistance to therapy. And so this is sort of the modularized pipeline that James in the lab has developed over the last sort of period. He tells me this is now redundant, and so there's a new approach coming out that we'll put out in the next couple of weeks. But essentially, you go through a multiplex staining of these tissue arrays. You go through an image processing. You identify your areas of necrosis, imaging artifacts, staining QCs. Protein data is quite different to sort of transcriptomic data. And then you do your clustering and identification of cell types. Over cluster, under cluster, and somewhere in the middle is where you probably have your real cell types. You run that past, 3 rounds of immunologists just to understand whether those cell types are actually real or not. And then all your sort of spatial feature analysis and comparative analysis. And so in this study, when we looked at the responders versus the non-responders, we can look at the individual cell types at a proportion level and contrast between the responders to immune checkpoint versus the non-responders. And we can see that there's this increased Treg population in the non-responder group. And so we can also do sort of neighborhood analysis, identifying the different neighborhoods that are different between your response group and your non-response group. And we see this macrophage mixed tumor phenotype that's enriched for in the responder group. And sort of more sort of distance-based statistics, we can start to look at proximities of different cell types next to each other at the protein level. So we can look and ask the question, is there an association of having 2 different cell types next to each other in individual patient samples, but then also extrapolated across the cohort? And we can see that the presence of Tregs next to monocytes, HLA-DR positive tumor cells next to macrophages, or Tregs next to CD8 cells is associated with response or resistance to second-line IO in the study. And so we're validating a number of these findings at the moment. And so what we've done is we're now benchmarking our multiplex IF data to pathology annotated H&Es as a ground truth level. And this is over, thousands of images of H&Es, where we also have multiplex data above those H&E data sets. And so what this allows us to do is, I'll skip through this slide, is it allows us to now, once we've got the metabolic, the enzymes against metabolic activity, we can now subset on every cell type here. So you've got, for example, where your tumor cells are, you've got metabolically active tumor cells as group 0, group 1, but then you've also got metabolically dim tumor cells here in group 2. The same can be done for CD8 T cells. So you've got metabolically active CD8s, metabolically dim CD8s. And this third group, when you go back to the imaging data, this is actually the CD8s that are infiltrating the tumor. So you can categorize and subtype every cell type using those functional markers over phenotype alone in sort of larger cohort studies. And so what we find is that the immune cells are generally found on the left of the seat map, the tumor cells on the right. And the key take home from this study was we've got this SCT2 glucose 6 phosphate dehydrogenase positive tumor cell group or tumor cell subgroup that is associated with a worse outcome to checkpoint inhibitor therapy. So it's not just about the immune context here, but it's also about the metabolic status of those tumor cells that's playing a role here. And so I'll go on to this head and neck cancer. So we do both tissue arrays and whole slides. Whole slides are beautiful. There's a lot more biology happening in them. And so this was a head and neck cancer patient who had a partial response to immune checkpoint inhibitor therapy, died after 14 months. So we had the pre-IO specimen, the surgical resection. We knew that there'd be heterogeneity. Our question was, could we look at it? Could we find it? So we stained a serial section adjacent to what the pathologist looked at with 101 markers, 103 initially, narrowed it down to 101. And so it revealed, and we modeled this on the hallmarks of cancer. So metabolic phenotyping, tumor immune phenotype, proliferation, stress, death, cell death, and structural and cell signaling markers. So sort of forming it on the hallmarks of cancer. And what we found were that these tertiary lymphoid structures, these mature TLSs were infiltrating tumor regions 4. They were absent in tumor regions 4, 2, and 3, which was interesting because we know that these tertiary lymphoid structures sustain an immune responsive microenvironment, but they're only localized in this tumor region 4. We could break this down and understand where all the other different immune cell types were, and also the structural components of this TLS, and that it was a mature TLS. When we looked at the RNA content and contrasted it to these normal germinal centers that are distant from the tumor, they're completely different. This is at the whole transcriptome level. And so we can see that these TLSs that are infiltrating the tumor have a very different mRNA profile compared to distant germinal centers, and pathways involved in interferon and so on are enriched for in these TLS components. And so when we broke this tumor down into subtumors, again, very gross annotation based on protein data, we find that the M1 macrophages are localized to the area where the TLSs are. The M2 macrophages, which are generally considered immune suppressive, are found in the purple region of the tumor. And so sort of on multiple dimensions, we can start to understand this partial response to therapy, the presence of TLSs in one region, which are mature, but also this M1, M2 polarity within the tissue. What was really interesting for us was when we added in the layer of metabolic activity over these tissues. And what we found was that tumor region 3 was metabolizing glucose a lot higher than tumor regions 4, 2, and 1. So G6PD was increased in tumor region 3, whereas tumor regions 4, 2, and 1 had increased expression for your pro-apoptotic markers. So in this one patient's tumor, you've got an area of sensitivity at the top and an area of resistance at the bottom, defined by your immune cell biology, but also metabolic activity, which is really interesting. So when we think about heterogeneity and resistance to therapy, this is the complexity of the challenge that we're trying to address, and we can now visualize that using spatial genomics and transcriptomics and proteomics. And so what we built was a spatial metabolic map. We've identified that this pentose phosphate pathway is enriched for in tumor region 3. You've got DNA damage and fatty acid metabolism on the periphery. And what we think is the invasive edge of this tumor is this edge here, which has increased expression of MMP9, whereas all of tumor regions 4, 2, and 1 are dead, dying, or necrotic regions. And so we can see that at the RNA level too, with increases of CXCL9, CXCL10 at the top of the tumor, and increases of CCL22 at the bottom of that tissue. And so you've got pro-tumor and anti-tumor activity in the same patient's tumor. And so we're building these and trying to impute some of this information back into H&Es and sort of trying to democratize these approaches. And so in the field, one of the challenges is most of the community is looking at PD-1, PD-L1 protein expression, but we actually wanted to know where are these actual interactions happening. And so we used this proximity ligation assay, which when you've got the receptor and ligand within a certain distance, you've got rolling circle amplification, and you can see the fluorescence signal. And so now we can contrast complete responders to checkpoint versus progressive disease or non-responders, and actually visualize the PD-1, PD-L1 interaction and not infer it from transcriptomic data, which is what the field generally does. And so what we see is that the staining patterns for this proximity ligation between the complete responders. Here, CR is a complete responder, PR is partial response, PD is progressive disease, SD is stable disease. But when you contrast the response that in the purple, you can see that the complete responder, the localization of the interactions is completely different to the PD patient, the non-responder. You've still got the interactions in the progressive disease patient, but they're at the boundary of the tumor and they're not really infiltrating the tumor. And so we've now scaled this study. We've got a preprint out on this, and we're applying it across larger cohort studies. This is in about 32 patients where we're showing a snapshot here. And so we can mimic this and look at it in skin cancer. Skin cancer is really sort of easy to profile over the course of therapy. I won't go into the major findings, but we can start to contrast over the course of therapy the same site biopsy between responders and non-responders. And we find that, you know, there are cell proximity differences between your baseline and your follow-up sample in skin cancer. And I'll end with some of the work in COVID-19, where we had these tissue arrays built in Brazil, COVID, flu, normal, and we ran multiple transcriptomic assays on these platforms, on these samples, and I've flagged a number of studies here. Our key take-home was we identified that IFI27, a type 1 interferon, was enriched for in COVID, was found in the lungs of COVID-19 patients when we contrasted it to control and flu. And this was conserved across multiple studies at the time. And so you get this peak in interferon, or this type 1 interferon, which subsides later on. We identified it using a multicellular readout, and then we wanted to localize this to the single cell. And so we looked at an independent cohort of patients across, so 40 patients across 2 TMAs and a normal control. And what we found was that this signal was coming from, and we ran the 1K Cosmics panel, we found that this signal was coming from the macrophages. We submitted this work. The reviewers came back and said that there are 10 different types and flavors of macrophages. Which ones are they? And so we went and looked at alveolar macrophages, interstitial macrophages, and because we had the plex level, we could identify these subsets and indeed show that it was found in alveolar and interstitial macrophages. And then Tony, similar to Ilya's talk earlier, got fed up with cell segmentation and said, what if we looked at this in a transcript sort of cell-free approach? And so he looked at the same cohort, 100 million locations, 100 billion transcript to transcript distances. And so you're essentially looking at sort of dots in space rather than containing them within a cell. What he found was that he could identify, you know, the major pathways, so interferon gamma, fibrosis, and so on, you know, pathways we know using cell-based approaches that we can find difference or perturbed in COVID-19. But what was interesting was we could also identify changes in oxygen CO2 exchange in red blood cells using a cell-free approach, and also perturbations in the nerve cells, which is a really small subset of what's found in the lungs. So these wouldn't be traditionally found using cell-based approaches. And so this preprint will come out in the next couple of days. And I'll end with this sort of crazy study where we had 105-year-old tissues, had a student spend a year in Germany and Austria collecting these, brought them to Brisbane, and we ran a whole transcriptome and about a 570-plex protein panel. This is the pathologist annotation of regions of exudate and pneumonia at the protein level and the PCA showing that what we can see is that the exudate and the pneumonia, the histology separates out on the protein data. That's the differential expression between two of these groups. The challenge now is to find normal control tissue for 100-year-old samples. This is the RNA data, the exudate in the red, the hemorrhage in the green, the pneumonia in the blue, and the transudate in the purple, but the transcriptome PCA there and the differential expression between two of these. RNA is a bit wobbly, but we've got a couple good samples there. This is what the protein data looks like on 105-year-old tissues. We ran a 21-plex, of which we have signal of across about 7 markers. And I'll end there and thank the lab that do a lot of this work, the patients that have contributed these samples to multiple studies, and I look forward to the discussion. Thank you.
Thank you, Arutha. You already won the challenge for the best virtual background by far. So now I invite all the panelists to join, to switch on your cameras and join the panel. I mean, we've heard amazing talks, and there was quite some discussion in the Q&A forum already. I would take some of the questions now to the panel because Arutha, you will not have the time to answer. So I think one question that came up again and again is kind of the robustness. Question to you, Arutha, across TMAs, because you have many samples, obviously, different qualities. Can you comment how technologies on the RNA, on the protein side, perform across those arrays, and do QC kind of metrics behave the same?
Yeah, that's a good question, Holger. It's something we battle with all the time. The reason we do tissue arrays is because we can traditionally get 50 to 100 samples on one slide, right? So the cost effectiveness of your spatial assays, the cost of that experiment is a lot reduced. We wouldn't be able to do 100 whole slides, for example, at whole transcriptome single cell due to the cost today. So there's a trade-off between running whole slides and then being able to build tissue arrays where you've cored sufficiently across that, where it's representative of the tissue at a whole slide level, but then you've maybe done 4 cores per block onto a TMA. We haven't done, 10 serial sections, same block, and looked at the integrity of the signal as we move through the block. Those, I think, will happen as the costs of these assays come down. For the moment, it's a trade-off between being able to screen versus being able to look at the really interesting biology of whole tissues, which is some of what Anna and Ilya spoke about. Because I think in the whole tissues, that's where all the exciting biology is. But then where sort of the challenges are around the costs in being able to scale that.
Thank you. And you and also Anna, you were showing like full transcriptome, or let's say 18-plex panel sizes. So what, in your experience, what did change moving from a 500-plex, 1,000-plex to a full transcriptome?
So on the one hand, obviously you detect a lot more counts and features in a per cell basis, which theoretically will give you a better representation of the complexity that you have on the tissue, right? So because we've profiled, for example, CRC in both assays, in the 1K plex and the WTX, right? So in the 1K, we're talking about 200, 500 genes per cell. And on the WTX is around 800, 1,000. So it's more than a lot more, right? Features also are quite also distinct, but the fact is, though, nevertheless, so you get more counts, you get more features. You can definitely profile more subtle states within each, cell compartment or cell lineage. That's for sure. But I still see this problem in which highly variable genes are not cell type defining genes. So we do not know yet how to determine what are the marker genes that we need to use to then perform clustering analysis with these types of assays. That's why it comes so handy to have the single cell references. Yeah,
and I think Arutha, you were facing different challenges, right? Because your sample was very old. Still, it turned out to be useful.
Yeah, so I mean, we had these virologists that we were working with during COVID, and so they'd been looking at, peripheral samples for a long time, and they got excited by being able to look at tissue. And so, crazy experiment, crazy samples, but I think we now have the technology it's pretty much proof of concept, right? And if we're getting protein data, and in some of the samples, RNA is pretty conserved, we have the whole blocks. If you cut them fresh and you're using sort of these probe-based methods, there is an opportunity to look at, tremendous cohorts of archival tissues, going back. We don't have to go back 100 years. It might be 10 years, it might be 20 years, but it's unlocking the archives and not having to prospectively collect these tissues.
No, fantastic. And my next question relates a bit to kind of the scale in terms of kind of areas to be profiled, numbers of genes, numbers of cells. So how do you see the, let's say, technology readiness level for the current state-of-the-art spatial methods to generate atlases of the healthy humans, so Human Cell Atlas project applications? That's probably more for Anna and Ilya. And Arutha, on your side, how much can that be already applied, pushed into the clinical research, clinical digital pathology context?
Ilya?
You want to kick it off?
Yeah. I can try to tackle this one. I think that's that's a real known unknown for us right now, right? So we we have these beautiful MERFISH cancer samples. We're able to outline the tumor boundaries, the stromal networks, the immunity hubs, and everything. And then we register it to H&E, and then we look at the whole H&E, and we're like, oh my God, we're getting like 1% of this giant thing, right? So in cancer, for sure, right? So in cancer, this is definitely like a huge problem, but it's a solvable problem potentially with technology and algorithms, right? One solution is obviously throw more money at it, sequence everything, image everything, and that's that's a limited scale. But I think there's some kind of technological middle ground where you can do a high-plex assay on key areas and then a low-plex assay across everything. I mean, the extreme thing is H&E, right? There's a lot of really great deep learning models that can predict gene expression just from H&E alone. And I'm always skeptical how well it's going to generalize from person to person, but within one tissue, within one slice, I'm actually pretty optimistic that you can actually generalize and sort of impute what xenium MERFISH would have looked like if you would have spent $200,000 imaging the whole piece of tissue. I think we're in an interesting situation where in cancer we have that problem. In autoimmune disease, we have this opposite problem where the treatments for, let's say, RA are getting better and better and better. And so you don't have people coming in with these giant, oftentimes with these giant inflamed joints, because they're already being very well managed to a certain degree with biologics, and we can't get these giant samples like we get in tumor. So we're actually faced with more of an unknown because we're getting small biopsies, which is great for patient care. But then we're always left wondering how much of the spatial heterogeneity have we actually captured of this patient. So I don't have a solution for that, but I think it's just it's an issue. So it's an unknown that I want to throw out in there. That depending on the disease, this issue of spatial heterogeneity, spatial coverage, how much do we know, how much do we not know, I think is very tissue specific, disease specific.
Anna, any thoughts? Are we ready for prime time?
I think we are getting ready in terms of the technology is getting really ready for it. Also, when comparing, some metrics that we get out of these data compared to, like the ground truth that could be the single cell, I think we are not far from that. So it shouldn't stop us, right, to really use it for building atlases. Now, the problem is what Ilya was saying. How much do we need to profile to really capture the complexity, the spatial complexity that we have in an organ? But the technology is ready or almost ready for it. Yeah.
But then ask me just one follow-up question, because you mentioned single cell as a ground truth, and I mentioned in my overview the different levels of complexity. But I think none of you really showed an integration of single cell data with the spatial data, right? So there are still computational challenges. So where are we in this kind of area, moving from single cell resolved atlases to spatially resolved and combining both informations together?
I can jump in also with that. I think I agree 100% with Anna saying that, right now the single cell is absolutely necessary in all of our studies in order to interpret the spatial cells. And this is touching on some of the questions that came up in the chat. I think this is largely because cell segmentation is so imperfect that if I have a T cell next to a fibroblast, that T cell is going to be like 80% T cell genes and then 20% or more fibroblast genes, because it's hard. It's hard to localize and very accurately call out these cell types. And then especially when I get into subtyping, right? I'm not, if I just do like regular clustering on spatial cells without integrating with single cell, I'm going to get 10 different T cell clusters, fibroblast positive T cell clusters, B cell positive T cell clusters, right? And so on. but not Tregs and TPH and CD8s and all of that stuff, right? And so, you know, how we overcome that now is with single cell. So hopefully as segmentation algorithms get better and better, we're going to lose our dependence on single cell. But right now, I think I'm actually very optimistic about the state of integration of single cell with spatial. We've been doing that for years now, and we get pretty robust answers, you know, robust alignment between the two technologies, even when we have only like 200 or 300 genes. And it becomes very clear, you know, you're kind of limited in the number of cell types you can call, but if you're careful about it, you know what the limits are, and it's actually, you know, a very useful tool right now.
And Anna, you mentioned the in situ typer, right, as an integration tool that can integrate and use single cell as a reference, but also work reference free, right?
Yeah, and that's just because we in this particular project we work with Cosmics data, right? So if we work with another type of spatial transcriptomics assay, then we can use another tool for that, which will be most adequate. But and that brings me also to the fact that the problem that we also have is that depending on the technique that you use, you need to tailor your whole computational analysis just based on that, which is fine, but we have to define how and what to use in each case, just because if not, you keep on pushing on some direction that is just not meaningful at all. But I also wanted to maybe emphasize shortly, because now and I totally agree with Ilya, right? So we need the single cell profiles as reference to make sense out of the spatial data. But if we say that, it might seem that, you know, and I said single cell is the ground truth, and then which leads to spatial technologies, which we love, like a sort of on a bad position. And I just wanted to maybe emphasize the fact that although that's absolutely 100% true, you also discover a lot of new biology just because you look at this data in space, right? Even if you are using single cell suspension profiles as a reference. So if you allow a bit of flexibility, you find new biology in there, either that you were not finding in the reference data set, or even that you would have overlooked in the reference data set just because it wasn't popping out as something special, which is just special because it's particularly distributed in space. So spatial is the way to go.
So we have to become open-minded to find new biology and new things in the data. And talking about challenges, and maybe going now backwards from the clinical side, Arutha, because obviously the clinical diagnostic context is one, but the clinical research context, it has very specific requirements, right? To scale that across many samples, you mentioned TMAs, is cost the main boundary really to apply such technologies at scale, or is it the resolution still where you mentioned the 101 protein panel, where you kind of come in with a stronger hypothesis?
Yeah, it's a great question, Holger. I'm just reflecting on what Anna and Ilya was saying earlier. I think, you know, obviously, you know, Anna's data is incredible, the whole transcriptome single cell, and as those technologies become mainstream and commercial, and that scales, which will happen over the next 12 months, I think it potentially becomes prime time for spatial, and obviously you need the backbone of single cell data, but I'll also sort of frame it in that ground truth fundamentally for clinical or translational work is your H&E, right? So you do need your pathology annotated H&Es. And so at a fundamental level, we should be scanning every slide we run spatial on with the, you know, same slide H&E, and just banking that slide, because within time we'll need to use that H&E. And I do see a question in the chat about, you know, how do we democratize these approaches? It's using that H&E, right? If we can impute this spatial data, you know, that costs thousands of dollars per sample into an H&E costs 50 cents to run, right? Any lab around the world can do that and scan that and upload that image. And so I think that's how this moves forward. And also just reflecting on SITSI last week is, Peter Sorger's talk on at 5 micron, we're only seeing 20% of the cell, right? And so to actually get the whole cell in its volumetric sort of density, we need to be at about 35 to 40 micron. And so we don't get that from pathology. We don't get that from histology labs. In a research setting, we can do that. But, then we have to think about, diffusion of antibodies through that thickness and things like that. So I think the cell segmentation tools and algorithms will improve, but are we missing a lot of the biology, or are we only seeing a small snapshot of it, or cross sections of, half cells and things like that at 5 micron? And it's, is that where, we potentially need to think about some of the 3D components to really be able to do proper segmentation? And I remember at AACR, there was almost a 4-hour session on just cell seg. And so lots of challenges for the field, but I think at a minimum, we need to be bringing in that H&E for all slides, be it TMAs or whole tissues, as we move it forward into the sort of translational clinical space.
I think staying with these challenges, you all mentioned cell segmentations to be not perfect these days, but it is obviously crucial to derive single cell resolve profiles, not from tissues. So in the future, do you see solutions coming that involve, let's say, 3D approaches, or this is a problem that cannot be solved with the kind of current approaches or current way we approach it these days? So it's solvable or not solvable?
I'm going to let Anna and Ilya answer that because I've tried.
We can do better. We can do better than we have now. Yeah, no, I don't think this is a 100% like solvable problem. Like we will never, I don't think we'll ever know. And when we look at a cell, whether transcript 100% belongs to that cell, and it wasn't called by mistake, and it wasn't localized incorrectly, or yeah. But I think we can do better and better. Yeah, I think more information definitely helps. 3D helps. Having multimodal, protein and RNA together, is more is better. Having more genes also is better, right? Because we can sort of build these gene programs from the data and say, okay, I'm not just going to try to call a T cell by this one gene, but it has to have these 50 genes. So that makes things more robust. And yeah, so I'm hopeful. I'm hopeful that we can do a lot better at cell segmentation. And I think that there's kind of a few things that doing better means. One is assigning transcripts correctly to the cells, and so we can just use vanilla tools like clustering to actually find fine-grained cell states and activation states. And the other one is also being able to distinguish noise from cells and being able to call the number of cells, right? I think we know that there's a bias in a lot of times we look at our macrophages and transcript-based segmentation methods are going to take our macrophages and fibroblasts and divide them into 3 or 4 cells often. And so we know that we're getting the cell numbers a little bit wrong too, right? So I think both in terms of cell counting and transcript assignment, we can do a lot better. And we will do a lot better, but it's not 100% solvable, I think.
Great. And let's do a bit of a wish list for the future. I think we touched Omics, but maybe Anna, if you kind of have Ilya and Arutha here, so what would be your wish list in kind of translating your technology into the clinical context, or to Ilya saying these are the computational tools that need to be developed, that need to be improved before we really can leverage the full power of spatial genomics? Sorry for putting you on the spot here.
No, all good. Wish list. So I think what we are definitely missing, but I don't know what to really. I mean, on the one hand, definitely, so cell segmentation is an issue. So how do we call a cell that's an issue? But we've discussed about that, so that's good. And maybe I will just add in there also as a part of the wish list, maybe to try to go beyond the single cell sort of mindset and just trying to reflect the single cells in space, but just try to go beyond that, because there's a lot more biology in a tissue than single cells positioned in a particular way. And this might also be a sort of a way to go around this challenge as well. Not only looking at cells, but only looking at molecules. And Arutha said or mentioned and showed actually that there are also some interesting tools for that, right? So just to use molecules and not try to use the cell as a unit. So that's one example, but also then tissue microenvironments, niches, whatever we want to call this, etc. So to really develop computational tools that will allow us to overcome the natural challenges inherent to spatially resolved data, which is signal bleeding just because cell segmentation is not perfect, or all of these things. That would be on the top of my wish list.
I can't agree more. My brain is still stuck with UMaps. So coming to me, doing single cells forever, it's like now really making this switch is not easy. So I love your answer. Yeah, go ahead.
And maybe for Arutha, so I don't know if we can think, if it makes even sense to think on that in that way, but and just because of what he was saying, right? So if we want to make it cost effective and to have some clinical relevance, we need to work on smaller tissue areas just to be able to profile a lot of donors, right? So, but in order, so how to decide what's relevant in each sample or in each patient, and would that maybe help not only to have like a whole tissue or a whole slide H&E, but to have some sort of three-dimensional representation, like very low plex, but a three-dimensional representation of that sample that we can then look into and define in which X, Y, and Z position we need to look at, and then scale that in numbers to really then implement it in a relevant clinical setting. So to define a strategy, to design the experimental part. I stop.
Yeah, it's a great point, Anna. I think what we see is what we try to do is to look at the extremes of response, right? So when you look at a complete responder versus a progressive disease patient, somewhere in the middle, your partial response and stable disease is sort of, you've got a mixed patterning, if you will. When you look at your two extremes, your complete responders microenvironment is very different to your non-responder, right? So somewhere between those two, I think there's a way that we can narrow down a strategy and how we do that. But most patients end up in that middle spot where the signal does get quite mixed. But we need, I think we can all work together to understand these complexities.
I think we can go on forever. But Ellen, I think it's time to wrap up, right? So it was a beautiful presentation, first of all, a great discussion. We should just continue offline or find a forum to keep on discussing. Thanks for joining today. Thanks specifically to Arutha for staying up late night. Time for more coffee or just go to bed. And with this, I will finish. I wish you all a good rest of the day, wherever you are, and looking forward to the next webinar focusing on spatial genomics.
Thank you,
Ellie, over to
everyone,
you.
for joining. Yeah, and Holger, thank you so much for moderating, and to all our speakers, thank you so much.